basics of the adaptive response. Okay, so we've talked a wee bit about this already. Need you to be able to differentiate between innate and adaptive immunity. Innate immunity is when it's the first line of defense, it's non-specific, immediate, and has no memory. Basically, an innate response, it just recognizes that something non-self is present. Okay? And to a certain degree, it might recognize that a bacteria is present. It might recognize that a virus is present. But the one thing the innate response cannot do is tell which virus or which bacteria or which fungi. And in that sense, it has no memory and it's non-specific. But it's essential because the innate response is rapid. So the innate response gives us the ability to try and do something immediately to reduce the infectious load. And while that's ongoing, this innate response, again, we need it because it's quick. But while that's going on, the adaptive response will start to kickstart off, will start to initiate its response. And the beneficial aspect of the adaptive response is it not only knows that there's a bacteria present or a virus present, it knows exactly what virus or bacteria that it is. And the advantage of that is that when we get an infection with, say, Clostridium difficile, for instance, the immune system knows to target that specific pathogen. And because it knows what specific pathogen is present, it can tailor its response to be extremely effective at targeting that pathogen and nothing else. Okay? But the important thing to appreciate is both have a role in the response to the response to an infection. The innate has a role because it's quick, it's rapid, and the adaptive has a role because in the end it's going to eradicate that pathogen in its entirety in a very specific and efficient manner. Okay, these are some of the cells that are involved in both. So as I said before, the adaptive is specific and it's tailored. Okay, and we'll talk a little about this later on, where the B cells can actually amend their gene to be more specific, to have a greater affinity against a pathogen. Because we also have to appreciate the pathogens evolve as well. They're going to change slightly. So the adaptive response gives us that opportunity to tailor a specific response. And one of the other advantages of the adaptive response is, as you can see in this graph, the first response is slow because we're just trying to find out what specific pathogen it is and then try to tailor our response according to this specific pathogen. And the problem with that is it takes time to do that. But once we've tailored a response, after that, if we get infected a second time, we've already did all the hard work. We've developed that tailored specific response. So if we get this infectious agent a second time, we don't need to do the specificity, the tailored response bit. We're ready to process the adaptive response. So that's the difference between the innate and adaptive response. And the receptors of the innate response are those pattern recognition receptors. Now what I'm going to move on to is what makes the adaptive response different. And the thing that makes the adaptive response different is B cells and T cells. And the thing that makes B cells or T cells unique or different is the B or T cell receptor. Every cell, every T or B cell in your body has its own unique receptor. Okay? Generally speaking, if I took 2 B cells out of your body and I looked at the B cell receptor in each, they would be different. But there's a reason for that. We need each B and T cell in our body to be able to respond to a specific pathogen. Okay? And the reason for that is that with all the pathogens that we're exposed to, we need to have a T or B cell that has the ability to respond to that pathogen. Okay? To allow that specificity. So, B cell receptors, every B cell will only ever recognize one pathogen, okay? If I take one B cell out of your body and look at its B cell receptor, say for instance it will respond against Clostridium difficile. That B cell will never respond to any other pathogen. It's like a process of division of labor. It has one job, to look after one pathogen. And there's another B cell to look after a different pathogen. Okay? And it's the same with T cells as well. They have, each individual T cell has its own unique receptor that will recognize one specific antigen or epitope of a pathogen. Okay, so let's just cover the concept of antigen. Antigen is anything that responds against, anything that the immune system recognizes. Now, one of the difficulties is some people consider pathogen to be some infectious agent. But we have to appreciate that we actually have self-antigen as well, okay? So self-antigen is proteins or components of our cells that make up our cells. The last thing we want is for the immune system to create a response against self-antigen, okay? And we have a process for that. But again, just to appreciate that antigen can be both self and non-self. They're usually large and they're usually proteins. And the other concept I need you to grasp is that, okay, we have antigens, but the specific part of the protein or molecule that is recognized by the immune system is called the epitope. So the epitope is the part of the antigen that's recognized. So for instance, here you can see that these this is an antigen, and they contain 4 different epitopes. So what that means is this antigen can be recognized by the immune system at 4 different locations within the antigen. Okay? And what will happen then is, when we create an immune response, there's the potential for 4 different B cells to produce 4 different antigens, 4 different B cells to produce 4 different antibodies, and each one of the antibodies will recognize a specific epitope. Okay? So again, each antigen has different epitopes present. And what you'll generally find is, okay, in this case, we have one antigen with 4 epitopes. Generally what you'll find is one of the epitopes will be more easily recognized than the other. And when that happens, that would be defined as the immunodominant epitope. That is the epitope that is most strongly recognized by the immune system. Okay? So that's the basic concept of what an antigen is and what an epitope is. Now, I'll relate to both antigen and epitope interchangeably, but I just need you to appreciate the difference between the two. So, how do B cells recognize antigen? B cells recognize antigen by directly binding to the antigen. Okay? And what I mean by that is they express their B cell receptors on the cell surface, and that gives the B cell the ability to directly recognize antigen using its B cell receptor by directly binding. Okay? And the main message from this slide is B cells do not require antigen to be presented. Okay? They directly bind it. Now, it's a different story for T cells. T cells will never be able to recognize antigen unless it's broken down and presented to it. And what happens is the antigen in question will be taken up into a cell, or will be present in a cell. It will be broken up, and the component or the epitope that the T cell recognizes will be attached to either MHC1 or MHC2. Okay? And that epitope then gets presented to the T cell receptor of the T cell, and that is the point at which a T cell can recognize antigen and also, secondly, become activated. So the T cell receptor binding the MHC is signal 1 for T cell recognition, for T cell activation. Okay? Signal 2 is when you get the attachment of B7 on the antigen presenting cell to CD28 present on the T cell. Okay? Two things you need for T cell activation, signal 1 and signal 2. Signal 1 is T cell receptor binding the MHC, and that'll cause a CD3 signal into the T cell to give it signal 1. Signal 2 is when CD28 binds to B7, and that will cause signal 2, and it'll become activated at that point. Okay? But back to the basics. T cells recognize antigen when it's broken up and presented to them. Okay? Now, this is generally, and this is a generalization. It will change, and we'll explain how it changes as we go on. But if it's antigen that's present in the cell, such as a virus, so an intracellular virus, the antigen will be broken up and presented on MHC1 to CD8 cells. Okay? Now, if it's an extracellular pathogen like a bacteria, what will happen is the bacterial antigen will be taken up by an antigen presenting cell, broken down, and the epitope will be attached to MHC2. And that will then interact with it'll interact with MHC2. Okay? And at that point, it will become activated. So that's the basics of antigen presentation. As a general rule, intracellular antigen gets broken up, attached to MHC1, and presented to CD8 cells. Secondly, if it's an extracellular pathogen, the antigen gets taken up by an antigen presenting cell, broken up, attached to MHC2, and presented to CD4 cells, to a CD4 T cell. Okay? That's that. So it raises the question of why do T cells need antigen presented to them? It's actually like a division of labor. We're going to use B cells to recognize antigens that are on the to recognize epitopes that are on the outside of the antigen. Okay? So here you can see this red epitope here. That's going to be recognized by a B cell. And it's one of the reasons why B cells can directly bind, because the binding site is on the outside, it's accessible. Now if we take a step to the side, in order for the T cell to recognize its epitope, it's trying to reach the blue epitope. Now you can see it's going to be very hard for the T cell receptor to directly get access to that epitope. And that's one of the reasons why a T cell is going to need the antigen to be broken down, because its T cell receptor simply can't get at the epitope that it needs. Okay? And here we are here. B cell has no problem using its B cell receptor to bind to its epitope, which is the red portion. But now we go towards where the T cell wants to bind to its epitope, and of course it can't get access. The epitope for the T cell is actually inside the antigen, and it's inaccessible. So the antigen presenting cell does its job, takes the antigen up, breaks it down to isolate the blue epitope, and at that point it attaches the blue epitope to MHC, and at that point, because that blue epitope is on the MHC, it's easy for the T cell receptor to interact with it. So that's the rationale why B cells don't need antigen presentation versus the rationale for why T cells do need antigen presentation. Now that we've cleared that up, we're moving on to the specificity. Just again to get across that every T or B cell in your body has its own unique receptor. Okay? And each T cell with its own unique receptor will only ever recognize one epitope. Okay? It's not possible for a given T cell to recognize two different pathogens. It's impossible. Its T cell receptor is only designed to recognize one epitope. Okay? Now, this, you've seen this before, but this is just to highlight the principle. Every immature B cell or T cell has the exact same copy of the gene for their B cell receptor or their T cell receptor. Okay? They all start the same. The way that they're able to obtain their own unique receptor is based on the fact that when they go to mature, they manipulate the B cell receptor gene or the T cell receptor gene in a way that it modifies it so that each cell has its own unique gene. Okay? So they start off with the same gene, but when they go to mature, they start to randomly change their B cell or T cell receptor gene. And when they do that, what it ensures is that after maturation, they all have different versions of that B cell or T cell receptor gene. Okay? And that's how they have different B cell or T cell receptor genes. And in terms of visuals, here you can see that each immature B cell has the same B cell receptor. Okay? Versus when they undergo maturation and they do their gene rearrangement, each individual B cell develops a different version of that gene. And here you can see Salmonella has changed, this B cell has changed its B cell receptor in a way that allows it to recognize Salmonella. Whereas this immature B cell has matured and changed its B cell receptor so that it can recognize E. coli, and so on, tapeworm and Clostridium difficile. Okay, so appreciate that. Every immature B cell starts with the same copy of the gene, then it changes the gene, and when it changes the gene, it gets a unique receptor. Okay? So, this is what happens. If we get a Salmonella infection, the only cell of those 4 cells that can respond and recognize against it is the Salmonella, is the B cell that changed its B cell receptor gene to enable it to bind and recognize it. The rest of them won't be able to recognize it because their new receptor just doesn't bind to it. Okay? In the case of E. coli, that T cell is the only one to recognize it, and so on. Tapeworm and Clostridium difficile. Okay? And it's the same with the T cell. Okay? Each T cell starts with the same T cell receptor gene, and then during maturation, it modifies it in such a way that it will have its own unique T cell receptor. Okay? So here we go again. Here we have a rotavirus infection, and the T cell that has differentiated in a way that will recognize the rotavirus will bind to it and recognize it where the rest won't. Okay, herpes. You want to recognize herpes, measles, and so on. So what I want you to get an appreciation for at that stage is that each individual B or T cell has its own unique receptor that will recognize a specific pathogen. And here's a group of B cells here, and you can see that each B cell recognizes its own pathogen, has its own unique receptor. And likewise, T cells have their own unique T cell receptors that will enable them to recognize the pathogen. Right. I hope you have an appreciation for that. Now we're going to move on to what T cells do what. Okay? So, if we have a viral infection, generally speaking, it will involve CD8 cells and macrophages. Okay? They're the cells that are going to kill virally infected cells. With a bacterial infection, what we're going to have is B cells, the production of antibodies, and phagocytes. Okay? Now, if we get a parasitic infection, we're going to have B cells, antibodies, and If it's a parasite, eosinophils. Now the specific response associated with parasites is not just an antibody response, it's IgE. Okay? So it's IgE mediated antibody response. And if we get a fungal infection, we will have a Th17 response. Now we'll have a Th17 response if it's at the mucosal surfaces, okay? But if it's within the body, say for instance in the kidneys or in the lung, in the kidneys or in the liver, it's going to be a Th1 response, okay? So there is a difference whether the infection takes place in the mucosa or inside the body. Okay? And for your own knowledge, the viral infection will be a Th1 response. Bacterial infection will be a Th2 response. Parasite infection will be a Th2 response. And fungal infection will be a Th17 or Th1. Okay, we've been through this before, okay. Endogenous antigen gets presented on MHC1 to CD8 cells. Exogenous antigen gets presented on MHC2 to CD4 cells. The one thing you have to also appreciate is that when antigen gets presented on MHC2, there's only specific cells that can do it, okay? Dendritic cells are the best at it. Then you have B cells. Then you have macrophages, okay? They're antigen-presenting cells. So there's only 3 cells that are professional antigen-presenting cells. B cells, macrophages, and dendritic cells. The thing to appreciate about MHC1 is it's expressed in nearly every cell in your body. It's not expressed highly in the nervous system, and that's another issue. But generally speaking, MHC1 is expressed in every cell in your body. But of course that makes sense because every cell in your body is at risk of getting infected with a virus. Okay? So, we've talked a little bit about MHC and this process of MHC presentation and its ability to bind to T cell receptors is called MHC restriction. Okay, should be aware at this stage of MHC1 and MHC2, MHC1 presenting viral antigen, MHC2 presenting MHC1 presenting antigen from within the cell, and MHC2 presenting antigen that has derived from outside the cell. Okay, now that's the very basics of adaptive immunity. Now, Now, in terms of the adaptive response and any question associated with the adaptive response, this is your, these are the series of steps. Antigen will be presented to CD4 cell to activate the CD4 cell. And as I said before, T cells need to be activated through signal 1 and signal 2. Okay? And at that point, the dendritic cell presents the antigen to the T cell. Okay? And this is the start of the immune response. Okay? Dendritic cell breaks down the antigen, presents it to the CD4 cell, and at that point we decide what type of immune response we're going to have. Okay? Are we going to have a Th1, a Th2, a Th17, or a Treg response? Now what dictates whether we do Th1, 2, 17, or Treg? What dictates whether we go down each of those paths is the cytokines that are produced by the dendritic cell. Okay? So by default, in some ways, the dendritic cell decides what type of response we're going to have. And the way it dictates to the CD4 cell what path we're going to have is it releases cytokines. So dendritic cell collects antigen from the infected tissue, brings it to the lymph node, presents the antigen to the CD4 cell, and at the same time releases cytokines, which are essentially like a set of instructions to the CD4 cell to say, you need to become a Th1 cell, or you need to become a Th2 cell, 17 or reg cell. Okay? So at that point, we've decided what type of T cell response we're going to have. The next thing we need to ensure is that we have sufficient numbers of that CD4 cell. Okay? You've got to appreciate that when we get an infectious agent and we present it to the CD4 cell that's responsible for recognizing that pathogen, it's actually present in very low numbers, which isn't going to be good enough. So we need to increase the numbers of that CD4 cell that's responsible for recognizing that pathogen. And the way we do that is through a process called clonal proliferation. And this process of clonal proliferation enables us to go from, say, one CD4 T cell to millions of CD4 T cells. Okay? So antigen collection, presentation, selection of the type of T cell response we want, and subsequent clonal proliferation of that CD4 T cell. The next step we need to do is, once we've got enough T helper cells, that is enough CD4 T cells, we now need to recruit and activate the cells that are actually going to kill the pathogens. Okay? And in the case of a Th1 response, it will be CD8 cells and macrophages. In the case of a Th2 response, it will be B cells and eosinophils. In the case of a Th17 response, it'll be neutrophils, monocytes, and macrophages, and in some literature you'll even see CD8 cells as well. Okay? I mean, the Th17 response is like a crazy alarm going off. It's just a case of we need a massive response because we've got a massively infectious agent. Okay? Now, the alternative is, if during this process we present self-antigen, what we need to do is cause this process to end, or to cause this process to differentiate towards Treg cells. Okay? Because Treg cells will switch off any aggressive immune response. Okay? So once the effector cells are activated, they will then once we activate the effector cells, we then need to increase the number of those effector cells. Because we're in the same scenario where we have small amounts of effector cells, and what we need to do is increase the proliferation of those effector cells to ensure that we have sufficient numbers. Right, that's a general synopsis. Now we're going to go into a bit more detail. If you get asked any question that relates to T cell, any adaptive response, this is your checklist. Okay? Anything that asks about Th differentiation, this is your checklist. Okay, so firstly we have antigen. First of all, we have antigen presentation to the CD4 cell. Then we have selection of the appropriate CD4 helper cell subset. Then we have expansion of that CD4 cell subset. And at that point we take a pause. We have enough CD4 cells. Now, next step. Let's try and get some effector cells. So what happens is we get activation of the effector cell. Then we need to ensure that we have sufficient numbers of effector cells, so they will generally undergo a process of clonal expansion. Once we have clonal expansion, we have sufficient numbers of effector cells. Once we have sufficient numbers of effector cells, then those effector cells can progress and go to the site of infection and destroy the pathogen. Okay? Once we've destroyed the pathogen, we need to switch off the immune response. Okay? So as a general synopsis is, get sufficient help through CD4, then get sufficient cells that will do the killing, and then kill the pathogen. Okay? Now, what we're going to go through first is an adaptive response to a viral infection. Now, because it's a viral infection, there are different ways of MHC presentation. MHC1 presentation will take place through, or will help to present viral antigen. Okay? And this is just an overview of the different ways in which viral antigen can be presented. Okay? First of all, you have a cell that's infected, breaks down the virus and presents it to CD8 cell. The second and third are associated with dendritic cell antigen presentation. Okay? Where the dendritic cell uptakes viral antigen from outside the cell and processes it and presents it on MHC1. That's one way that it can be presented. The second is where you get exchange of viral antigen between two dendritic cells. Okay, an infected dendritic cell and an antigen-presenting dendritic cell. Now, this is the process for antigen presentation. Okay, for a virus. What happens is I'm not going to go through it now, we'll go through it in the next presentation. Okay, but just to highlight that for endogenous antigen presentation, it goes through this process. Okay, using the proteasome and subsequently attached to MHC1 and presentation to the surface. Now, for the presentation of exogenous antigen, it's done through the exogenous pathway. Okay, and that's this pathway here. Okay, it's taken up, and through the use of invariant chain and clip and Caspin S, it's attached to MHC2. Okay, that's the basic concept. All I need you to get is MHC1 is used to present antigen that exists outside the cell, and the exogenous pathway is used, MHC1 is used to present antigen that exists within a cell, and the exogenous pathway is used to present antigen that exists outside the cell. Now, this is where it gets a little bit confusing. Okay? Up to this point, I've talked about the fact that viral antigen is presented on MHC1. Are we all in agreement with that? Yeah? But when we get to this stage, there's a slight change in plan. Okay? One concept I need you all to get is that every adaptive response to any pathogen requires help. It requires CD4 T cells. Okay? Now, in order to activate CD4 T cells, what type of presentation do we need? MHC1 or MHC2? MHC2. So you see the problem here? We want to get help for a viral infection, yet generally speaking, viruses are presented on MHC1. Yeah? But in this scenario, what we need is to present viral antigen on MHC2. Is that clear, yeah? And I'm going to take you through that process. So, the dendritic cell has collected antigen from the site of infection. Okay? And this is where it differs. What has happened here is there are viral particles outside the cell. Okay? So what you have is a viral infection, and the cell has lysed and released the viral particles. Does that make sense, yeah? Because those virus particles are outside the cell, the dendritic cell takes it up and presents it on MHC2. Okay? Because the dendritic cell sees it as exogenous or antigen that exists outside the cell. Okay? And it presents it on MHC2. which is useful because we need to activate CD4 T cells. So this dendritic cell comes along with the virus presented on MHC2, and the difficulty we're going to have here that the dendritic cell is going to have is it arrives at the lymph node. Now when it arrives at the lymph node, there are literally millions of different T cells, okay? And the job of the dendritic cell is to find the CD4 T cell that has a T cell receptor that can recognize, in this case, the flu virus, okay? You see the difficulty there? So this dendritic cell arrives with the flu virus attached to its MHC2, and it has to find, in amongst all these cells, the one cell that recognizes the flu virus. But inevitably, it finds that specific cell. It presents the viral antigen to the CD4 T cell, and at this point, it's a naive CD4 T cell. Okay? And at that point, when the T cell receptor binds the MHC, it gets signal 1. Okay? That's signal 1. And the thing that actually gives the T cell signal 1 is the release of CD3. Okay? So that's signal 1. The antigen has been presented to the CD4 cell. The next step we need is for both cells to bring together B7 and CD28. At that point, we get signal 2 and the cell is activated. Okay, don't forget, for a T cell to become activated, it needs signal 1 and signal 2. Now, we have one activated CD4 T cell that can respond against the flu virus. But the problem we have is that's not enough. We need more than one cell. So what happens is, well, the first step that needs to happen is the dendritic cell needs to send instructions to this CD4 T cell what type of response it needs. So the dendritic cell comes along, presents, activates the CD4 T cell, and then releases cytokines to instruct the naive CD4 T cell, you need to differentiate into a Th1 cell. And the way it gives those instructions is through the release of IL-12. Okay? So IL-12 is a cytokine that instructs a naive CD4 T cell to differentiate into a Th1 cell. Okay? And the reason in this case that the dendritic cell has released IL-12 is because it knows that this particular component is an intracellular pathogen. Okay? So, it doesn't select the Th2 cell, it doesn't select Th17, and it doesn't select Treg. It selects Th1. So at this point we've had antigen presentation, CD4 T cell activation, and now selection of the appropriate Th response, which in this case is a Th1. So we have a CD4 T cell that has been activated and instructed to become a Th1 cell, and that Th1 cell will now produce IL-12. Okay, the dendritic cell will produce IL-12 and instruct the CD4 cell to differentiate into a Th1 cell. Okay, so the appropriate helper cell has been selected. Now, we have one CD4 T cell that has differentiated into Th1 cell that is able to recognize the flu virus. Now the problem we have is there's only one of it. Next thing that needs to happen is clonal expansion. And the way clonal expansion happens is the activated Th1 cell will secrete interferon gamma. Okay, and this process is called autocrine stimulation. The Th1 cell produces interferon gamma and binds on receptors in the Th1 cell. And that causes the Th1 cell to proliferate exponentially. And what we have at this point is sufficient CD4 T cell number. Okay? So we've got sufficient CD4 T cells. Now, that's the first phase. Let's pause there and appreciate how the CD4 T cell became activated, how it differentiated into Th1 cells. Okay. Now, that is one event. Another event was also happening simultaneously. Okay. What has happened is the dendritic cell has also taken up the antigen and presented it on MHC1. Okay? And it's presented it on MHC1 to the CD8 cell. Okay? And it's the same scenario as before. The antigen presenting cell presents MHC1, and the binding of MHC1 to the T cell receptor causes signal 1 on the CD8 cell. And B7CD28 causes signal 2 in the CD8 cell. Now we have activation of the CD8 cell. Okay? And it's the CD8 cell that's going to kill the pathogen. Now, we have activation of the CD8 cell, but we don't have sufficient numbers of it. So, it's the same process as before. But what happens here is this is where the help meets the effector cell. So the help that we got earlier, the Th1 cell, releases IL-2, which causes the activated CD8 cell to start proliferating. Okay? And again, that step is called clonal expansion. So what we have now is sufficient numbers of help and sufficient numbers of effector cells. And at that point, we have all the tools to eradicate the pathogen. So what happens then is the activated T cell now needs to get to the site of infection and kill the infected cell. So the T cell leaves the lymph node, goes through the tissue, and arrives at the site of infection. Now, one of the reasons that the CD8 T cell knows, one of the reasons it knows where to go is through a process called chemotaxis. Okay? The infected tissue will be releasing cytokines that will enable the CD8 cell, the activated CD8 T cell, to go immediately to that site. Now, here we have the CD8 T cell arriving at the site of infection. Okay? And as it arrives, it needs to know what cells are infected. One way that it is able to get to the rough location of the site of infection is the fact that the infected cell is releasing interferon. So the CD8 T cell then moves towards the site of infection, and it knows what cells are infected because the infected cells will be expressing MHC1. And at that point, the CD8 T cell is able to use its T cell receptor to recognize the antigen presented and subsequently interact with the cell. Now, at that point, we've got the right CD8 T cell to the site of infection. It's recognized the infected cells, and there's only one thing left to do, which is to kill the infected cell. And it does that through the release of perforin, which create pores in the cell, and granzyme. Okay? And here you see the CD8 cell has just killed the infected cells, but the one thing we have to appreciate is there's still a mess to be cleared up. Okay? The CD8 T cell has lysed the cells, but there's still potentially viral particles present. And that's where the macrophage comes into play. The macrophage will then come in behind and phagocytose all the debris. All the debris, including the viral components, which may still be intact. And it does that through the release or use of nitric oxide. Okay? Nitric oxide is a radical species, so the macrophage will use nitric oxide to destroy the structural components of the virus and also the structural components of the dead cells. Okay? And it increases nitric oxide production through the use of interferon gamma. Okay? So folks, that is the steps involved in eradicating a virus from an adaptive point of view. Okay, now we're going to move on to what happens when you get an infection that involves exogenous bacteria such as exogenous antigens such as bacteria and parasites. Okay? But the important thing to recognize is your checklist is the same. Okay? Irrespective of whether it's a viral infection, a bacterial infection, a parasitic infection, irrespective of what infection it is, this is your checklist. Okay? So once again, we have the process of antigen presentation. In this case, the antigen presenting cell takes it up, breaks it down, and presents it on MHC2. Okay? Through the exogenous pathway. So, here we have the dendritic cell has arrived from the site of infection, has broken down the antigen through the exogenous pathway, and presented the antigen to the CD4 T cell that recognizes that pathogen. So MHC2 interacts with the T cell receptor and you get signal 1. So in this example, we have a salmonella infection. And in this case, the salmonella has been broken down and presented on MHC2 to the CD4 T cell that has a receptor that can recognize salmonella. So once we have presented it, you get signal 1 and signal 2 as before through T cell receptor and also through CD28. And you get the CD4 T cell becomes activated. Now at this point, as before, the dendritic cell will release cytokines to instruct the naive CD4 T cell to differentiate into either Th1, 2, 17 or Treg. Now because this is a bacterial infection, what will happen is the dendritic cell will release cytokines that will tell the naive CD4 T cell to differentiate into a Th2 cell. Okay? And at that point, it will release IL-4, which will instruct the naive CD4 T cell to differentiate into a Th2 cell. Now, at that point, we have the appropriate helper cells selected. Next thing we need to do is get sufficient numbers. And as before, we have this process of clonal expansion. And again, it's an autocrine mediated process in which the Th2 cell releases IL-4, and IL-4 binds the IL-4 receptors on the Th2 cell, which causes mass proliferation or clonal expansion. Okay? And at that point, we have sufficient Th2 cells present. Okay? Okay, so we have sufficient CD4 cells, sufficient Th2 cells, sufficient numbers. So we have sufficient numbers of Th2 cells. And at that point, we need then to activate the effector cell. Okay? And this is how it happens. So again, pause at that point. We have sufficient numbers of Th2 cells. Okay? Put that to the side. What was also happening at that point was the effector cell was being activated. And the way the effector cell, in this case the effector cell is going to be B cells, the way it's activated is through direct binding of the B cell receptor to the antigen. Okay? So the Salmonella activated the B cell by binding to its B cell receptor, and at that point the B cell gets signal 1. Okay? Now, in order for the B cell to get signal 2 to get activated, it needs to take up the antigen that's bound to the B cell receptor, break up the antigen, and present the antigen to the corresponding Th2 cell. Okay? So the B cell takes the antigen up, breaks it down, presents it on MHC2 to the Th2 cell that's just been activated. And one of the reasons we need this to happen is that in order for the B cell to get signal 2, it needs to bind CD40, the CD40 ligand on the Th2 cell. And at that point, the B cell is activated. Okay? So, we have the activated effector cell, and once again what we need is sufficient numbers of that effector cell. And the way that's achieved is through the release of cytokines by the helper cell, the Th2 cell, in the form of IL-4 and IL-5. And when the helper cell releases IL-4 and IL-5, it'll bind the receptors on the B cell and cause clonal proliferation. And now we're in a position where we have sufficient numbers of B cells that recognize Salmonella. Okay? Now, we have sufficient numbers of B cells, but this is where it gets a little bit complicated. Okay? So we've just had a B cell recognize an antigen and become activated. Okay? And that B cell has a B cell receptor that's specific to Salmonella, and it binds fairly okay to the Salmonella antigen. But what the adaptive system wants to do is to further fine-tune the B cell receptor to ensure that it has not just an okay affinity for the antigen, but that it has a super affinity for the antigen. And this is how it happens. The activated B cell enters the follicle in the lymph node, and when it enters the follicle, it stops producing its B cell receptor. Okay? And when it stops producing its B cell receptor, it's then at that point referred to as a centroblast. Okay? And the reason that it stops expressing its B cell receptor is it's going to start tinkering with its B cell receptor again. Okay? It's going to start playing about with its B cell receptor gene, and in some of these cells, it's going to change it in a way that increases affinity to the antigen. And in some of the cells, it's going to change the B cell receptor gene in a way that its B cell receptor has less affinity for the antigen. Okay? So each one of these centroblasts changes its B cell receptor gene in a slightly different way. Okay? And when it changes in a slightly different way, it then starts to re-express its B cell receptor. And as I said before, some of these B cell receptor, because it's tinkered with its B cell receptor gene, actually won't have a good affinity for the antigen anymore. And that's fine. But some of them will have an even stronger affinity for the antigen. And at this point, what happens is a process of natural selection. What happens is you have a competition. Each one of these B cells is in competition to bind to the antigen presented by the follicular dendritic cell. Because what the follicular dendritic cell is doing is presenting the Salmonella antigen, and whatever B cell has changed its gene to create a B cell receptor with a stronger affinity for the antigen will be able to compete and bind to the antigen presented on the follicular dendritic cell. And the reason that's important is if you're one of these B cells that isn't able to compete effectively and isn't able to interact with the follicular dendritic cell, it doesn't get a signal to survive. Okay? If you're one of the B cells that changed its B cell receptor in a way that increases its affinity for Salmonella and has managed to compete and interact with the follicular dendritic cell, it gets a signal to survive. And what you have at the end of this whole process is the isolation of a single clone that has an extremely high affinity for the pathogen, in this case Salmonella. And that cell wins the prize to deal with the infection. So this cell here has got a really high affinity, got the signal to survive, and it's now ready to eradicate the pathogen. But something has to happen before that happens. The B cell will then interact with the T follicular helper cell. And the reason it needs to interact with the T follicular helper cell is it needs to be told whether it wants an IgM, an IgA, an IgG, or an Ig response. Okay? Now, at this point, once this naive cell has been activated for the first time, the first time this happens, the B cell will always express IgM. Always. Okay? And the whole point of interacting with a follicular helper cell is to instruct the T cell, actually, on this occasion, we don't want IgM. It's an infection that has taken place in the gut, and therefore we want an IgA response. So, the follicular helper cell instructs the activated B cell to change from IgM to IgA. Okay? If it was a parasite, it would tell it to change from IgM to IgE, because you need Ig response for a parasite. Okay? But the general point is the follicular helper cell will instruct the B cell what type of antibody response it wants. And the reason you would switch from IgM to IgA, for instance, is because IgA is particularly good at reaching the gut and dealing with infections in the gut, whereas IgM generally is good at dealing with infections in the blood. Okay? So that's the rationale. Whether it's IgM or IgA will not change its affinity for the pathogen. Okay? When you change from IgM to another isotype, it just changes where the antibody is going to arrive to deal with the infection. So, the B cell has class switched, and at that point, the B cell, the activated B cell, will either become a plasma cell or a memory cell. A plasma cell is a cell that produces incredible amounts of antibody. Okay? Whereas a memory cell will not produce antibody, it'll actually just remain as a memory cell, so that if we get infected with Salmonella again, we'll be ready to respond rapidly. Okay? So that's the synopsis of that. Now that we have the B cell, how is the B cell going to eradicate the infectious agent? As I indicated before, the plasma cell will produce antibodies, and the antibodies will be specific to that particular pathogen. So here we have a Salmonella infection. The plasma cell releases antibodies, and what happens then is the antibody will attach to the Salmonella, because its Fab region has a specific region that will recognize Salmonella. And the important thing to be aware here is that an antibody has a Fab region, which is this part here, okay? And that's the bit that recognizes the antigen. The FC region, which is here, doesn't recognize the antigen, but it has a very important role in the immune response. Okay, so here, the Fab region is attached to the pathogen, and sticking out is the FC region. And the reason it's important for the FC region to stick out is so the phagocytes can bind to it. So that when the phagocytes bind to the FC region, it can then take it into the cell and destroy it. Okay? So here we have the antibody that has attached to the infectious agent. And here we have a phagocyte. And the phagocyte has specialized FC receptors. These FC receptors then are able to attach to the FC region of the antibody and basically phagocytose the whole complex and then destroy it through respiratory burst and enzymatic processes. It then moves on, gets to the next, and does the same, and so on. Okay, folks, so that's how a B cell will eradicate a bacterial infection. Now, in terms of a parasite, okay, here we have a worm. The thing that makes this different is the antibody will be IgE, okay? And the antibody will then bind to the worm, okay? And again, the FC receptors will stick out, the FC portion of the antibody will stick out. But the cells involved in this process are eosinophils and mast cells. And eosinophils and mast cells have these little granules that contain a range of lytic enzymes. And unlike the neutrophil, which took the infectious agent into its cell and degraded it, in this scenario we can see that the worm is actually going to be too large to take into the cell and degrade it. So what happens is these eosinophils and mast cells use their FCE receptors to bind to the IgE that's attached to the worm, and once it binds, it releases its lytic enzymes outside the cell, and they will then subsequently degrade the worm. So you're talking about eosinophil peroxidase, ribonuclease, deoxyribonuclease, and a whole range of major basic proteins and other, okay? And it will degrade the worm and eradicate the infectious agent. Who said that? Why is that not happening? Yeah. Well, that's part of the challenge is, it's a good point, but one of the say, for instance, there's loads of different worms in our environment. That particular worm you're talking about has evolved a way to evade the immune system. So the immune system doesn't know it's there. Yeah, so that's essentially what happens in that scenario. Okay, is that us?